Hey everyone, it's Carolyn, and today I'm going to talk a little bit about cellular respiration and how cells produce ATP from glucose, specifically the steps that actually involve creating ATP, NADH, and FADH2. Cellular respiration sort of combines a bunch of different processes. Glycolysis, turning pyruvate into acetyl COA with the pyruvate dehydrogenase complex, which I talked about in another presentation, and the citric acid cycle, and then taking the NADH and FADH2 and putting them through the electron transport chain and oxidative phosphorylation. So glycolysis is when glucose is transformed into two molecules of pyruvate. So we see this 6-carbon structure of glucose becoming this 3-carbon pyruvate, and it makes 2 molecules of that. In this process, 2 net ATP are created. So as we can see in these 2 steps here, step 1 and step 3, which involve hexokinase and phosphofructokinase, ATP is actually used and formed into ADP. What happens is, so glycolysis needs ATP to begin. So these 4 ATP created in steps 7 and 10, 2 of those will actually be used for the next cycle of glycolysis in steps 1 and 3. So the 2 steps that are actually forming glycolysis take 1,3-biphosphoglykosylate, and using phosphoglycerate kinase, create 2 ATP and creating 3-phosphoglycerate. So we're removing one of these phosphate groups here. And in steps 9 to 10, or I guess step 10, phosphoenolpyruvate becomes pyruvate by removing this phosphate group here and transforming ADP into ATP. So we see we have 2 net ATP and we have 2 NADH here in step 6 using glyceraldehyde 3-phosphate dehydrogenase. So we're removing hydrogen atoms and putting them on NADH as we remove a phosphate group again. So when we're transforming pyruvate into acetyl CoA, we're going to be doing this twice because we have 2 molecules of pyruvate. Now when we talk about the whole pyruvate dehydrogenase complex, the enzyme we're going to be focusing on is pyruvate dehydrogenase, or E1. As we take the acetyl group and put it on to coenzyme A. So a carboxyl group is removed from pyruvate, releasing carbon dioxide here. NAD is reduced to NADH, and CO2 is also released. And the acetyl group is transferred to coenzyme A, resulting in acetyl CoA. And since we have 2 pyruvates, this is going to happen twice, giving us 2 NADHs. Now the citric acid cycle is where most of our ATP, NADH, and FADH2 is going to be coming from. So this will run 2 times for each molecule of acetyl CoA, the same way that pyruvate dehydrogenase complex ran twice. The acetyl group moves from acetyl CoA to oxaloacetate to form citric acid. Citric acid is a 6-carbon molecule, so we're going from a 4-carbon molecule to a 6-carbon molecule. And as we move through the cycle, we're reforming oxaloacetate back to the 4-carbon molecule. So in this process, we get 2 just ATP here because we're running it twice with succinate thiokinase. Now, most of our ATP coming from the citric acid cycle is going to be in the form of NADH or FADH2. So as we can see, the first step we're going to see NADH forming is when isocitrate becomes oxalosuccinate using isocitrate dehydrogenase. We'll see some of the structures on my next slide, so I'm just going to move through the steps that are forming NADH. When we get alpha-ketoglutarate moving to succinyl CoA in this step here, so that would be steps 4 and step 6. And then we have 1 more molecule of NADH with malate dehydrogenase. Now, as we can see, these are all dehydrogenase enzymes that are creating NADH because we need these hydrogen atoms to create NADH. We need to reduce NAD+. Now, we also have one molecule of FADH2 being formed right here, also with succinate dehydrogenase. So we're going from succinate to fumarate right here, and we get an FADH2 here. So in total, we have 2 ATP, 6 NADH, and 2 FADH2 molecules being formed in the citric acid cycle. Now, as we can see, I've also included the structures here. It makes it a little bit easier to see what's going on. So once the CO2 gets removed, we're also going to be losing this H here. And since we're creating a CO double bond, we're going to get another H off of this carbon. So we're losing 2 hydrogens there. That's going to be happening anywhere we're getting NADH and then FADH2 as well. So what we're doing is essentially we're getting rid of carbons to reform this oxaloacetate here. So another acetyl CoA can come in, and then the acetyl group can be added to oxaloacetate to create citrate, and the Krebs cycle or citric acid cycle can continue. Now, how do we convert NADH and FADH2 into ATP? We do that in something called oxidative phosphorylation and the electron transport chain. So as we move through this electron transport chain, the electrons from NADH are being moved, essentially pumped through the mitochondrial matrix into the intermembrane space. And up in here where we have all of these protons, we're getting a lot of energy. Now we need to take that energy and make it ATP. So as these electrons move down the electron transport chain, more and more protons are being pumped, and now we have a high positive charge in here compared to the matrix. Now this high positive charge, we use ATP synthase, which is an enzyme that creates ATP from ADP. So as we can see here, using this high energy, we use ATP synthase to create ATP, and that'll give us the energy that we need in our cells. So the total net ATP created, so in glycolysis we see we have 2 ATP. For each molecule of NADH in glycolysis, and this is specific for glycolysis, 1 NADH is going to give us 1.5 molecules of ATP. So we get 3 ATP from the 2 NADH molecules, which gives us 5 ATP total for glycolysis. Now for the pyruvate dehydrogenase complex, we have 2 NADH, and these NADHs are going to give us 2.5 molecules of ATP for each NADH. So here we have 5 ATP. Now in the citric acid cycle, we have 2 ATP, just right, straight ATP, 6 NADH, so 6 times 2.5 is 15 ATP, plus 2 FADH2. Now FADH2 actually enters the electron transport chain further down the line, so it's going to have less energy and therefore give us less ATP. So here, FADH2 is 2 times 1.5, which is equal to 3. So here we have 20 ATP, and as we can see, that gives us 30 total ATP. Now I should note that for pyruvate to become acetyl CoA, just in general, oxygen is required, and it's also required for the citric acid cycle and obviously oxidative phosphorylation. Now oxidative phosphorylation and the citric acid cycle will use oxygen outright. The pyruvate dehydrogenase complex just needs to happen in the presence of oxygen. So we need all of that to happen to get these 30 total ATP. I hope this was helpful. If you have any questions for me or if you disagree with anything, please let me know.